let's go ahead and get started. Today is Wednesday, October 7th. We are going to do the same thing as we did on Monday. Lecture is going to be bone physiology and lab is going to be our group presentations. You guys did a great job with that last time, so I assume today's groups will as well. I love all of the information and detail you guys are giving, and so this is really great. But again, remember, we really want to focus on the bones and bone features because that's what the test is going to be on. We have 3 more assignments. Our Unit 8 and Unit 9 review are due on Monday and on Wednesday. And then we have that 30-point skeletal review. Right now I've got it due on Wednesday, but I'm thinking Wednesday we still have bones and bone features to go over, so I may move that. Again, I figure we're online, might as well take advantage of it. So I may move it to Friday, possibly. I will think about that. But that unit review, that skeletal unit review is going to be available today so that hopefully you can start looking ahead and start thinking about that leading up till the exam. I don't want to have it due on the day of the exam. I don't like having assignments due on days when we don't meet, but I don't want it due on the day of the exam because I want you to be able to see the key, to see what you got right and what you got wrong. And those keys are only available after the assignment is turned in. So I may think about that, and I may change the due date for that so that we can finish our bones and bone features, because especially the lower leg are some of the trickier ones. All right, but that's the game plan. Where I want to pick up though is where we left off last time. Last time we were working our way through our lists of bones and bone features, and we had done that for the skull, but we had not had a chance to finish it for the the bones and bone features of the skull, but we haven't had a chance to finish the skull feature. So I want to pick up where we left off on that, and then we'll go into lecture and go from there. All right, so any questions before we dive into this? All right, so, oh, one other thing that I wanted to show you. We were talking about this in the last class, and this was the best picture that I could find of this. I remember we were talking about the superior nasal concha and its location, very posterior and very superior. And remember, when we look at it through the front of the skull, we're not able to see all of the concha. But here is the best view I was able to find from the posterior view. Notice this bone feature, which isn't one of the bone features that's on your exam for this, is the internal nares. This is the bony opening at the back of your nasal cavity. And if you look, you can see here are the 2 inferior nasal concha. Here are the 2 middle nasal concha. And over here on the right side, you see just a hint of it. This down in here, and again, you wouldn't see it on this side because it's a little too dark, but tucked down in there is where that superior nasal concha is. So if you have the skull and you tilt it just the right way, you can actually look down and up into that nasal cavity, and you can see all 3 of those conchas and how they are there to filter the air and produce that turbulence to swirl it around. So I did want to start by showing you that, but then I wanted to pick up where we left off, and that was working our way through the skull features together as a group. We had just done the orbit of the eye. How many bones make up the orbit of the eye again?
7.
7, excellent, right? What were they?
Frontal, maxilla, zygomatic, lacrimal, ethmoid, sphenoid, and palatine.
Excellent. So frontal, maxilla, zygomatic, lacrimal. And then on the inside, we have the sphenoid here. We have the ethmoid, which is over here on the wall. And that little bit of the palatine that sneaks up.
Pardon?
There.
Your mic is not muted. Oh, sorry.
No worries. So here is a slightly different view, which gives us a better view of the inside. So this is where I wanted to pick up. So again, as we were talking about, this out here is the zygomatic bone. This up here is the frontal. This down here is the maxillary. And here we get a really good view of that lacrimal, and we can actually see that hole that is formed by the lacrimal bone, which is where that lacrimal duct drains our tears into the nasal cavity. This also allows us to clearly see here is the ethmoid right here. It's the ethmoid. And then we can see our sphenoid here. And then, like I said, this little bump that sticks up in the back, that is the palatine bone. So those are the 7 bones that make up the orbit of the eye. All right, questions on that? All right, the next skull feature, and a couple of the skull features we're going to talk about next are openings of the skull. Now, these are skull features because they are not a hole in a single bone, right? The foramen ovale, for instance, was a hole in the bone. What happens here is you have one bone that has a notch in it. You have another bone that has a notch in it. And when you put those 2 notches together, you get a hole in between them. One of the things that is going to help you to identify these on the exam, well, there's really 2 things that's going to help you to identify these things on the exam, is one, the wording. There are a lot of holes that we see when we look at the bottom of the skull, like something like this. And typically the ones that are skull features are more irregular in their shape, because what you have is you have a part of one bone and a part of the other bone, and they come together. So notice, if you look closely, you can see there's this opening here. Anyone know what that opening there might be? It's kind of oval in its shape, foramen ovale, excellent. And there's a tiny little hole next to it, which would be the
foramen.
Tiny little pin prick or even spine-like opening.
Spinosome.
Yes, foramen spinosum, excellent. And remember, those 2 are right next to each other. That's going to help that. Maybe this one will be a little easier. Here we have an opening that tunnels through the bone, an opening that tunnels through the bone. That is the carotid canal, right? But notice here we have this very irregular shaped hole, right? And the same thing is happening over on this side as well. This opening here is the jugular foramen. So again, you have this irregular opening because you've got a notch of one bone and a notch of another bone coming together. And what are the 2 bones that are coming together to form this jugular foramen? What bone is this on this side?
Occipital.
Occipital. And then what would this bone on this side be? Here we see this big bump of it and the carotid canal that passes through it, and a broken spinous process right there, and the stylomastoid foramen. So what bone does that make this? Zygomatic process.
Temporal.
Fossa. Yeah, temporal bone, excellent, right? So notice it's the temporal bone and the occipital bone that comes together. Notice we see this really nicely from the underside of the skull, but we can also see this on the inside as the skull as well. So again, this irregular shape opening right here is the jugular foramen. There's one other thing that I want to show you. Again, you got to get used to identifying these holes from both points of view. And one of the things that always helps me to orient myself as I'm looking at this, and again, it's a little more challenging when you don't get to hold this in your hand, is let's go back to the underside view for a second. Notice here, and I'm going to change colors for them. So let's do, this is the jugular foramen, right? And then this right here is the carotid canal. Notice on the underside of the skull, these 2 openings are right next to each other. But what's interesting is the carotid canal, that hole penetrates through the bone and projects anteriorly, whereas the jugular foramen goes between the bones and projects posteriorly. So while on the underside, these 2 are right next to each other, if we look at the inside of the skull, this is that opening. Oops, there we go. This is the jugular foramen where it's opening here because it's come back. And as I mentioned, this right in here is where that tunnel comes out and comes out here. So notice the openings. We see the same thing here. It tunnels through the bone and comes out right there, right next to this very rough, jagged appearing opening. And so they're very far away from each other on the inside of the skull because they project in opposite directions. So you want to use these relationships. The spinosum is always next to the ovale. Those are an easy landmark that's easy to find, right? The foramen magnum is an easy landmark to find. Find these easy landmarks, and from there, you should hopefully be able to find your other openings and identify those. All right, questions on that? All right, the next irregular shape opening you're responsible for is the inferior orbital fissure. Remember, the superior orbital fissure is a bone feature. It was a bone feature of what bone?
The sphenoid?
Sphenoid, excellent, excellent. However, this down here is the inferior orbital fissure, and it is a skull feature. And what bones are a part of this skull feature? Excellent. Zygomatic bone helps to form a little bit of it over here. What else? What bone would this be? What bone forms the wall over here? Not the ethmoid. You're right, the maxilla is one of them, but the maxilla is actually this bone over here. This is the maxilla right here. So this purple bone that happens to help to form the superior orbital fissure and has the optic canal through it, what might that be? Not the temporal.
Sphenoid?
If only I knew what bone had the optic canal and this pure orbital fissure in it, I might be able to figure out what bone this purple bone was.
The sphenoid?
Sphenoid, there you go. So it's the sphenoid, the zygomatic, and the maxillary. Now, I did see someone say the palatine, and you are kind of correct. We do know the palatine kind of pokes right here, but I think the palatine more comes up into the inferior orbital fissure rather than really forming a part of it. So I would really say it's just the three bones, just the sphenoid, the maxilla, and the zygomatic. So those are what form the inferior orbital fissure. Now remember, we said the superior orbital fissure was a big opening where a lot of blood vessels and nerves and things like that can pass through. But there isn't anything that passes through nothing major that passes through the inferior orbital fissure. It's just a space that forms from the way these bones are growing together. Which brings us to our third opening that is a skull feature. And that is this one right here. And this one right here. This one is a nice obvious one because they're close to the midline. And they're very rough, very jagged in their appearance. All right? They almost look like they were torn or they were lacerated. And in fact, that is where it gets its name. What do we call this torn looking opening?
Foramen lacerum.
Yeah, foramen lacerum, lacerated, torn, right? That's what these are. This is so jagged looking because there are, again, three bones that come together to form this opening. One of them is this bone right here. What bone is that? It's associated with these condyles right here and associated with the foramen magnum right there, the occipital bone. Excellent. The next one is this bone over here that has the carotid canal passing through it and actually comes out at the foramen lacerum. So what bone might that be? Temporal. And the third bone is this bone right here with these wing-like extensions that happens to have the foramen ovale and the foramen spinosum in them. Sphenoid. There you go. Excellent. So those three bones come together to form this jagged opening that is the foramen lastum. And notice this can be seen from the outside. And remember, as we talked about, that carotid canal starts outside and tunnels towards the foramen lacerum. Whereas when we look on the inside, basically the opening to the carotid canal comes out right here where the foramen lacerum is. So again, this is the foramen lacerum. So get used to identifying these things from both points of view. I appreciate when you look at the bottom of the skull in particular, there's a lot of openings and it can get a little tricky and challenging, but there's a couple key things to remember when you're doing this. One is just start looking at the relationships of things. Like we talked about, come on, there. The foramen ovale is next to the foramen spinosum. We have the big obvious foramen magnum. We know we have that hole. We don't see it right here, but we know it's going to tunnel underneath the condyle. That is that hypoglossal canal. We know we have our carotid canal, which is tunneling through that there. We have this nice big obvious foramen lacerum. And then we have these irregular shape that we don't see great from this point of view, but we know they're here, jugular foramens. So you need to start working on the relationships of these things. Oh, and we even have our stylomastoid foramen, which we see really nicely on this one here, and this one over here as well. The other thing that's going to help you on this exam is, again, I will be precise on my wording. On the exam, I will have, you know, an arrow pointing to this space right here, and I will ask you to identify the skull feature. Even if you're not 100% certain what that is there any chance that the correct answer for that is the foramen ovale? No, because the foramen ovale is a bone feature, and I asked you for a skull feature. The same way, if I ask you to identify what this bone feature is, there's no way the answer can be the jugular foramen, because it is a skull feature. So if I'm asking for a skull feature, I will ask for a skull feature. If I ask you on the exam for a skull feature, there is only, right, however many this is, right, 12, 14 different possible answers that could be there. These are the only things that are skull features. So if I ask you for the skull feature on the exam, this is one of the things you should be giving me. All right. All right, let's finish off the skull features.
Whenever you're ready.
I'm ready now. All right, excellent. So next one are the cranial fossas. As you look inside the skull, as this is the cranial cavity formed by the cranial bones. But if you notice, it's kind of compartmentalized into three distinct regions at three different depths. We have this very shallow indentation right here in the front. We have a more intermediate indentation, really kind of two indentations here in the middle. And then we have this really deep indentation here in the back. So how do you think we identify these three indentations? Anterior, middle, posterior, cranial fossas, right? A fossa is an indentation. So we have our anterior cranial fossa, our middle cranial fossa, and our posterior cranial fossa. And the other nice thing about these is when identifying them, each one is comprised of three different bones. What are the three bones that form the anterior cranial fossa? What bones do you see as you look in here? Frontal, excellent. Ethmoid. Yeah, this one's got a great cribriform plate. Not so much the temporal, but what bone would this be right here? With the optic canals.
Sphenoid.
Sphenoid. There you go. Perfect. Those are the three bones that form the anterior cranial fossa. What are the three bones, types of bones? Because again, some of these are paired. What are the three types of bones that form the middle cranial fossas? Excellent. Sphenoid again. What else?
Ethmoid.
Ethmoid is way over here in the front, so I wouldn't say ethmoid. Sphenoid is this part here. What helps to form the lateral walls? There you go, the temporal. And what would be above the temporal, forming the lateral walls of this space? Parietal, excellent. Perfect. So those are our three bones that make up the middle cranial fossa. And that brings us to our posterior cranial fossa. Occipital, that's an obvious one, excellent. What else? What forms this raised ridge right here? Temporal. And parietal, there you go. Perfect. And there you go. Those are the three fossas and the three bones that make up each of the three fossas. Questions on that? All right, excellent. All we are left with then is the first joints we are going to be introduced to. Joints are where bones come together, and all of the bones of the skull form a very special type of joint known as a suture that holds them together. Or I should say all the bones of the skull except one. What is the one bone of the skull that doesn't form a suture?
Mandible.
Mandible, right? Which is why we are able to move it, right? But every other one, a suture is a special joint, a special non-movable joint of the skull. Every single joint of the skull, every single suture has a name. Are you responsible for all of them? Well, if only I had a list that I could look at to be sure. No. How many are you responsible for? 4. There you go. Exactly. So only 4. So that's not too bad. And as it turns out, all 4 involve the parietal bone. So that makes it easy as well. What's the first one on your list?
What? Oh, I'm paying attention.
What is the first one on your list?
Lambdoidal.
There you go, lambdoidal. Excellent. Anyone know what lambda is? It's a Greek letter, right? Good old lambda, lambda, lambda. Baby sheep, I like it. No, lambda is a Greek letter. It basically looks like an upside down V. Well, lo and behold, here on the posterior part of the skull, we have an upside down V-shaped bone feature, and that is the lambdoidal suture. Notice it is the suture between the occipital bones, the occipital bone and the 2 parietal bones. So this suture, someone already said baby sheep, they stole it, Elena. It is that upside down V between the occipital bone and the 2 parietal bones. That is the lambdoid or lambdoidal suture. The next one is the coronal suture. Coronal suture, we can kind of see from this view here. Coronal, if you think of the coronal plane of section, like a frontal plane of section, that would be along this line here. So notice the coronal section is between the frontal bone and the two parietal bones. While we're here looking at the superior view of the skull, we can also see the suture that goes right along the midline in between the two parietal bones. What might that be? There you go, sagittal suture. So we see that really nicely there. So again, all of these are pretty simple and straightforward in where they get their names. The last one is a teeny bit tricky, but I'll teach you how I remember it. The last one is the Squamosal Suture. The Squamosal Suture is this suture right here between the parietal bone and the temporal bone. So basically this suture right here between the parietal bone and the temporal bone is what is known as the Squamosal Suture. Now it is actually called the Squamosal Suture because what happens is the parietal bone comes down. And again, that term squamosal, meaning squamous, meaning flat, as we saw in our tissues, has a big flat surface to it that comes up. And then the temporal bone comes up and has a big flat surface for forming that joint. So this joint is formed by these two big flat surfaces that come together. So squamosal, squamous, again, one is the noun, one is the adjective. Same thing with lambdoid and lambdoidal. Both of those are acceptable. Lambdoidal is acceptable. Squamous, squamosal, both of those are acceptable as well. So yeah, both of those are acceptable. So it's called squamous, squamosal, because of this long flat surface of the joints that come together of them. All right. However, the way I remember it is if you look at it, the temporal bone has all this hunk and junk down here at the bottom, all these bumps, all these squiggles, all these parts, the ear hole and the mastoid sinus, and then the big flat part at the top. Squamosal means, squamous means flat. So it is the joint that is formed by the flat part of the temporal bone. So that's how I remember it. The squamosal suture is formed by the flat part of the temporal bone with the parietal. And there you go. So notice for all of these, the sutures, the fossas, even the orbit of the eye, you don't, the jugular foramen, the foramen lacerum, the inferior orbital fissure, for all of those, you just need to know the bones. There aren't any bone features for those you need to know because you didn't learn those, right? Zygomatic arch, you need to know the bone features, right? Perpendicular plate forming part of the nasal septum, you need to know that. The hard palate, those you need to know because those are on your list. But notice all the rest of these, you just need to know the bones. All righty. Questions on that? All right, excellent. What that means then is that we are now caught up. We have finished all of our bones and bone features and skull features that I wanted to cover for our in class last time. So now we can pick up where we were supposed to be, and that was back into lecture. So any questions before we get started? All right, perfect. Thank you, Haley. Thank you for providing that information. All right. Any questions before we get rolling? All right. So we were working our way through our bone physiology and starting with how you make a bone. And as we talked about, there are two ways to make the bone: endochondrial ossification and intramembranous ossification. So now that we've made the bones, our goal is going to be to grow the bones. And we said there are two ways to grow the bones. And how are the two ways to grow the bones? These are the easy questions. There we go. Length and width. Perfect. Excellent. So let's start with length first. Where does the bone grow in length? Well, careful. Epiphyseal plate, in the epiphyseal plate, absolutely. Right? Let's take a look at this and exaggerate this. Right? So here we've got our dino bone. And again, we know here in the metaphysis is a region that we call the epiphyseal plate. And what type of material is that epiphyseal plate made up of again?
Hyaline cartilage.
Hyaline cartilage, excellent. So let's take that and magnify it and really exaggerate it. Here will be our epiphysis. And let's move that up to there. And then down here, we will, perfect, draw our diaphysis. So obviously I'm exaggerating this tremendously, but if we were to focus in, and so it's not quite to scale, but if we were to look at this in the microscope, which we'll do when we look at the pretty pictures from your book, you can see that this epiphyseal plate actually contains 5 distinct regions. And these 5 distinct regions, not surprisingly, play a role in the growth of the bone in length. Now, the first region, and I'll draw this in purple because it amuses me to do so, is what is known as the resting zone. With a name like the resting zone, how active do you think it is in the growth process?
Not very active.
Yeah, not exactly. This region is not involved in the growth in length. Again, it's not that it's dead. It's not dead. This is living hyaline cartilage. It's all hyaline cartilage. But it's not active in the growth process. Instead, it acts as an anchor, where its job is basically just to hold the epiphysis in place. So again, it's going to be hyaline cartilage. It's going to have lacunas. These lacunas are going to have chondrocytes sitting inside of them. And it's not going to be involved in the growth process.
Is that like the perforating fibers that anchors the periosteum to the bone?
I see where you're going with that. I guess sort of, although I would say the periosteum does have those Sharpie fibers that helps to anchor it and hold it in place, act as an attachment point. So it definitely is similar in that fashion. Yes, absolutely. Where basically it just provides some structural integrity. So yes, I do like that analogy. That works. All right. So this is our resting zone. This is what it looks like here. And then, hold on. What just happened? Sorry. Okay. You guys see that now, right? Yes. Can you guys see the whiteboard? Okay, perfect. Excellent. All right. So the next region is what is known as the proliferating zone. Orange, I don't want that big. In this proliferating zone, what does it mean to proliferate? Yeah, if you're making more of, right? Proliferating, give birth to, making more of, right? So here in this region, what we have is rapidly dividing chondrocytes. as we know, the chondrocytes inside of the lacunas are able to divide. And when they divide, making two cells, we know those two cells are going to start making matrix. And as they start to make matrix, they are going to expand the tissue. And that's exactly what's happening here. Here we are expanding the tissue by making new matrix. What kind of growth did we call this again? Where we are expanding the tissue from within it?
Or length,
No, what was
growth
it?
in length.
Yeah, well, we are growing it in length, yes, but what is this actual growth process where we have the expansion of the tissue from within?
Endochondral membranous, intramembranous.
No, remember that's how we made
Interstitial
bone.
growth?
Say again?
Interstitial growth?
Interstitial growth, excellent. This is our interstitial growth. Remember when we talked about interstitial growth, that's how we made the cartilage model to make a bone. But we also said this was going to be how we grew a bone in length. So here we are using our interstitial growth. When this occurs, what ends up happening, since these cells are rapidly dividing and expanding and moving across against each other, what it looks like is we get this huge stacks of cells that look like stacks of coins, because all these cells, and I'm just going to draw the lacunas here, all these lacunas are being formed. The cells are constantly dividing. They're constantly moving away from each other. And we are getting the expansion of the tissue rapidly within this region. And after all, as this tissue expands, the epiphysis moves farther from the diaphysis, and mission accomplished. Right? The bone is longer. Yay. Boom. Just like that, we're done with the process. All right. End of story. We've reached our goal? No. Because is that what our bones look like? Do our bones look like they just have a tiny piece of bone in the center of them, and then the whole rest of it is all cartilage at the ends?
No.
No, of course not. But we have accomplished our goal of making the bone longer. The problem we have now is, yes, we've made the bone longer, but now we must convert that cartilage into bone. And lucky for us, we know exactly how to do that, because we did it in our endochondral ossification. Think back to when we made that long bone. We started with cartilage, and then we converted that cartilage into bone. What was the first thing that we did when we converted that cartilage into bone?
Blood vessel.
Say again?
The blood vessels.
Nope. Before the blood vessels could grow in, something else had to happen. What was that?
Mesenchymal cells turned into osteoblasts.
True, that's what happened on the outside, but what happened on the inside? What happened on the inside of our diaphysis? Before a blood vessel could grow in, we had to make a space for that blood vessel. And what did we do to make the space for that blood vessel? Well, maybe this will help. This next region is known as the Hypertrophic Zone.
The chondrocytes are going to die?
Exactly.
We had to grow first.
Exactly. Perfect. Excellent, excellent, excellent. You guys are absolutely correct. Hypertrophic. Trophic means what?
Growth.
Growth. And hyper means?
Over.
Over, increased. Exactly. So remember, the first thing that happened is our chondrocytes get big. And as our chondrocytes get big, their lacunas have to get big, and the matrix within it thins and hardens. And when that matrix thins and hardens, oh, so let's hold on, let's cheat. Put my big, large chondrocytes inside of this. And as you guys pointed out, when that matrix thins and hardens, our chondrocytes end up dying. When they die, what they leave behind is just basically a calcified matrix known as the calcification zone. So we have that hardened matrix cavity that forms. And as you guys pointed out, and the reason I used red is that here the blood vessels grow into the area. And as blood vessels grow into the area, they bring new cells and new signals, new chemical signals. So let's draw some quick blood vessels growing in here. Blood vessel growing in, blood vessel growing in. And as those blood vessels grow in, what color haven't I used? I haven't used blue. And my, oh, hold on. I can cheat. This is the one nice thing about the whiteboard. I can adjust my picture. See, I meant it all the time. Excellent. And then that leads us to our ossification zone, where now our osteoblasts and osteoclasts break down the calcified matrix and make bone. And in that way, we first, so we have, if you think about it, you have this epiphyseal plate. All right, so let's say it looks like this. We have this epiphyseal plate, this big. And then what happens is in the proliferating zone, we expand the matrix and make it larger. But then down here at the bottom, we convert some of that into bone. And then we expand the matrix and we convert it to bone, and expand the matrix and convert it to bone. Notice what's happening is my bone gets longer, but my growth plate always stays the same size. And so in this fashion, I can get from the tiny little baby to the big grown adult that I am. And that is exactly what happens. Now, as always, I've done an amazing job drawing this, but let's look at the pretty pictures from the textbook that show the exact same thing. Again, this growth and length occurs at the epiphyseal plate, but then the plate closes. We'll talk about that in just a second. And there are 5 zones to the epiphyseal plate. So here is what it actually looks like under a microscope. And we can clearly see these 5 distinct regions. So as we talked about, first up here, we have our resting zone. Again, the resting zone is not dead. There's just no active growth that is taking place. It is just hyaline cartilage, being hyaline cartilage, acting as that anchor for the epiphysis. Then we have our proliferating zone. Notice in this region, we have these many tiny cells looking like stacks of coins because they're rapidly dividing and pushing away from each other, rapidly dividing and pushing away from each other. So this is where we are getting our interstitial growth. This here is where we are getting our length. This is where the bone gets longer. using that interstitial growth. And that's perfect because that's what we want. We want the bone to get longer. The problem is it's making it longer with cartilage. So what we need to do is then convert that cartilage into bone. But we've already learned how to do that. All right, the first thing that has to happen is our chondrocytes have to get bigger. And notice as these cells get bigger, there is much less matrix in between them. And so we have these highly active large cells that now are having trouble getting their oxygen, now are having trouble getting their nutrients, and they are going to end up dying. And when they die, what they leave behind is just a hardened calcified matrix where our blood vessels can now grow in, bringing new cells, bringing new chemical signals, and allowing us to break down that matrix with osteoclasts and replace that matrix with bone using osteoblasts.
Is this kind of like the healing process? Because, you know, short people sometimes will break their bones and try to make themselves longer, taller.
So you are correct. So Gattaca was an excellent example of that. Great movie if you haven't seen it. It's a very old movie now, but an excellent movie about genetic modification. Yes, one of the things that they will do to try to increase people's length is that they will break the bone, put a spacer in, and then allow the bone to heal, and it expands the bone that way. It is an incredibly grueling, long, and painful process, but you can sometimes get an inch or two of height doing it that way. It isn't a very pleasant way to do that. And we will actually talk about a healing of the bone. And as it turns out, it does heal in a similar fashion, right? No, that type of growth you can do anywhere along it, because again, you're purposely breaking the bone in the diaphysis and trying to extend the diaphysis. So you would do that before the bone plates. One of the things that they will do with certain types of, let's do this. We only grow in length while our epiphyseal plates are open. Some forms of dwarfism occur because the epiphyseal plates close too rapidly. So one of the things they will do with those individuals is they will give them things like human growth hormone to try to increase the bone growth before that epiphyseal plate closes. And like I said, sometimes with those types of hormonal supplements, they can get as much as 4 inches in height. And I know 4 inches doesn't sound like a whole heck of a lot, but when you're talking about the difference between, 3'6" and 3'10", it's a huge, huge difference. Well, yeah, I guess it might sound like a big difference to us too, but if you're talking about a difference between someone who's 3'6" and someone who's 3'10", those 4 inches are a huge difference. But that does bring us back to our original problem. If I am expanding the cartilage and then making bone, and expanding cartilage and making bone, expanding cartilage and making bone, and these processes are always occurring at the same time, then my bones should get longer forever. Why is it that my epiphyseal plate closes then? What changes, what modifies this process and causes the epiphyseal plate to close?
Chemical signals.
And what chemical signals in particular? You're absolutely correct.
Hormones.
Hormones. And what hormones?
Follicular stimulating hormone.
Not a bad guess, but no.
Growth stimulating hormone.
Human growth
Parathyroid.
can help this, but think of it this way. When do most people start sprouting in height?
Puberty?
Puberty. So it's estrogens and androgens that have an effect on this. What happens is estrogens and androgens increase bone formation. Estrogens and androgens stimulate our osteoblasts so that they make more bone. So we get a growth spurt as a result of that. But the other event that occurs is now what happens, if you think of your growth plate, right? We make a little bit of cartilage, and then those osteoblasts, because they're more activated by the sex hormones, makes more bone. And then we expand the cartilage and it makes a lot more bone. And we expand cartilage and it makes a lot more bone. And notice what's happening is my epiphyseal plate is getting smaller and smaller. I make cartilage, and I make a lot more bone. I make cartilage, I make a lot more bone, until eventually I make cartilage and it ossifies all of it. At that point, I don't have cartilage anymore. I have a thin line of compact bone. And what do we call that thin line of compact bone?
Epiphyseal line.
The epiphyseal line. Excellent, excellent. So one more question to put it all together. Both androgens and estrogens stimulate the osteoblasts, but do they both stimulate them equally? No. Well, I'm asking the question, so obviously the answer should be no. So which one has the stronger effect on the osteoblasts? Androgens?
Androgens.
Androgens. I have 2 votes for androgens. Everyone thinks androgens? Well, let's think of this. Across the world's population, whose epiphyseal plates close faster, so typically on average are shorter than the other sex?
The girls, estrogen.
Females. The reason females on average, now again, even in a class as small as this, is every single male taller than every single female in this class? No, even though we've never even been in the same classroom together, I can guarantee with 100% certainty that's not the case. So obviously there's fluctuations, dramatic fluctuations, but as a general trend, females tend to be slightly shorter than males because their epiphyseal plates close faster. Osteoblasts are much more sensitive to estrogens, and there's 2 huge impacts to that. One is that both males and females reach puberty and they start making all of these sex hormones. And again, the females' osteoblasts are more affected by the estrogens, so their epiphyseal plates close sooner, so they typically tend to be shorter. But then also let's think long terms. Both males and females reach puberty and produce large amount of estrogens and androgens, and that continues until death.
Menopause.
No. Males keep producing androgens until death. They stop producing androgens, they stop producing sperm about 15 minutes after they die. Same thing for females?
No.
No.
They reach puberty, start producing estrogen, and then what happens?
Menopause.
Menopause.
Menopause, absolutely. Menopause is that point where, from a biological standpoint, a female becomes useless. And at that point, no. But what happens is
Is your wife listening?
Oh, she knows not to listen to me. No, but they reach that point of biology where they're no longer capable of producing offspring. But the impact for the female is there's a huge drop in estrogen levels after that. And part of the problem with that is, as we know, estrogens stimulate osteoblasts to make bone. So if you have a drop of estrogen levels, then suddenly your osteoblasts are not being stimulated as much. Your osteoclasts are still chomping away at the bone matrix, but we do not have as much bone matrix being made. So as women age, especially once they reach menopause, what can become a major concern for females?
Calcium.
Process.
Yeah, osteoporosis. Osteoporosis is that loss of bone mass, that loss of bone density, right? The problem is grandma can drink a whole cow's worth of milk. If she's not able to stimulate those osteoblasts to deposit that calcium into the bone, she's going to start losing bone density. And so one of the things that they do with individuals who have osteoporosis is they will actually give them estrogen supplements to help to boost that bone density and to hope to make that in there. And yes, I was teasing about the biologically irrelevant. But yes, and IVF is, again, amazing stuff. All right, excellent. So yeah, that was just a joke. But again, the point was to emphasize those differences in the hormone levels. All righty, and there you go. We have now grown our bones in length using that interstitial growth, looking at the 5 zones and seeing how that occurs. Any questions on that? All right, excellent. So what we need to do next is grow the bone in width. But because we were a little behind and had to do those the skull features, this is a good natural stopping point. So I want to go ahead and stop here. we'll take our first break here. It's 8:55, so let's come back at 9:10. And at 9:10, we will pick up by talking about how to grow the bone in width. And I will start the recording again at that point. All right. Any questions on that? All right, excellent. I will meet you guys back here in 15 minutes. All righty. Hopefully we're all appropriately caffeinated and ready to get back to it. All right. So let's switch gears again to here and talk about our growth and width. Now, again, because we want to be thinking in terms of similarities and processes, to grow in length, we use interstitial growth. What growth process are we going to use to grow in width?
Intramembranous.
No, again, remember, intramembranous is how we make bones. So we had two growth processes that we talked about. One was interstitial growth. Ah, there we go. And the other is appositional. Appositional growth, remembers, we're adding new layers on the outer surface. So how did we do that on our cartilage model? Anybody remember where that occurred? How were we able to add more layers to the outer surface?
Periosteum.
Yeah, on our cartilage, it was in the perichondrium, right? Because the perichondrium houses those mesenchymal cells, but you absolutely got the right idea. Here, our growth in width is going to occur in, or really under, let's say under, the periosteum. All right, so let's start with a really simple example. Here is our bone. And as we know, that bone has a periosteum on the outer surface. I'm going to draw it once, but again, this is where things get a little tricky. So All right, we have that periosteum on the outer surface, and we know that that periosteum houses those mesenchymal cells. We know those mesenchymal cells can divide to produce new osteoblasts, and the new osteoblasts can make more bone. So I'm going to cheat and redraw my bone over here. And so we know what's going to happen is an osteoblast is going to form. That osteoblast is going to surround itself with matrix and mature into an osteoclast. And that's going to occur everywhere around our outside of our bones. So another one's going to form, and another one's going to form, and another one's going to form. And in this fashion, we could produce a layer of matrix that goes around the entire circumference of our bone. And what would we call that type of layer of matrix that went around the entire circumference of the bone?
Particular?
Well, no, circumferential, right? Lamella. We would produce a circumferential lamella, and as a result of that, our bone would be wider. Again, mission accomplished. We know that the periosteum is still there, and so we know it's capable of producing another layer on top of that. And then we know it's capable of then producing another layer on top of that. And we know it's capable of producing another layer on top of that. And notice just that simply, just that easily, I'm making my bone wider. So at its core, that's all we're doing. We're adding new layers of matrix on the outer surface, and that is going to make our bone wider. Right? Under the periosteum, our mesenchymal cells become osteoblasts. Those osteoblasts make matrix. They make a layer of matrix that's going to go all the way around the entire circumference of our bone, and our bone gets wider. However, we have an issue. We have looked at a cross-section of a long bone, and does it look like a tree, right? You can just cut through the bone and count the rings and tell how old the person is. Is that what it's going to look like? No. We do know that there are a few layers of circumferential, but is the entire bone just circumferential lamellae? Is that what the entire mature bone looks like? Just hundreds and hundreds of layers of circumferential lamellae?
No.
No,
There's
it's not what it looks
separate.
like, right? So remember, not only is this process going to grow the bone in width, but remember we said it also is going to be how we form our osteons. So we need to form our osteons with this process as well. And that's how this is going to be different. We can't just cut through the bone and count the rings and tell how old you are. We also need to form the osteons. And the reason we don't get these perfect circumferential lamellae, these perfect circumferential layer of matrix around the entire bone, is because bone is vascular. And that's really the key. What ends up happening here is the blood vessels get incorporated into the bone. And that is how we get our osteons. That is how we form that organizing structure of our compact bone. So let's start over again. I want to leave the words up there at the top, so I'm just going to try to erase all of this stuff. Come on, there, and there, and there, and there, and there, and there, all of that. There we go, excellent. All right, and what I'm gonna do is I'm gonna increase the size of this. So what I'm gonna do, I need to cheat and move this over here and cheat and move this over here for a second. This is the edge of our bone, all right? So down here, we have our medullary cavity. Down here. And we know, and I'm only going to draw the teeniest bit of it, but we know that on the outer surface of our bone here, we have our periosteum with the mesenchymal cells. So let's add some labels so we don't forget any of this. So we have our periosteum with our mesenchymal cells. And like I said, this is our medullary cavity. And we also know this medullary cavity is lined with the endosteum. We have an endosteum in here as well. So let's go ahead and draw that. And we know that endosteum houses mesenchymal cells and some other stuff as well. All right. So as I said, the basics of this process is very straightforward. Mesenchymal cells divide, and when they divide, they form our osteogenic cells. Right? Those are our unipotent stem cells that form our osteoblasts. So what we have here is an osteoblast that forms. Once that osteoblast forms, it makes and surrounds itself with matrix. And when it surrounds itself with matrix, it becomes an osteocyte in a lacuna. Right? Really no new information there. So this is going to surround itself with matrix. And now it's in a lacuna. Bingo. All right. And again, this is going to happen right here next to it as well, and right here next to it as well, and right here next to it as well. And so in this fashion, we add a layer of matrix on the outer surface of the bone. Questions on that? Excellent. You know how much I love my stunned silences. Perfect. Excellent. So here is where things get interesting. Remember, The periosteum is on the outer surface of this, containing those mesenchymal cells. But remember, the periosteum also contains blood vessels. And so out here in our periosteum, and I'm not going to draw the periosteum, but I will draw the blood vessel. Out here is a blood vessel in the periosteum. And so what happens as our osteoblasts are being formed, as our osteoblasts are making, are making layers of matrix, what ends up happening is they respect the boundaries of the blood vessel. And they don't build matrix where the blood vessel is. And then another layer forms, and we respect the boundaries. And then another layer forms, and we respect the boundaries. And what ends up forming here is what we call these periosteal ridges. And these periosteal ridges form around blood vessels. Now, once we get past the blood vessels, these ridges start to elongate until the periosteal ridges are actually going to fuse together. Basically forming a cavity around the blood vessel. Now, remember, on the outer surface of this is still, there we go. On the outer surface of this is still that periosteum. But guess what? That periosteum was also lining this space as well. And when the two edges come together, they basically pinch off the periosteum inside that cavity. Of course, this periosteum, if it's inside of a cavity, is it really a periosteum anymore? No, it becomes an endosteum. But guess what? That endosteum still contains mesenchymal cells. And that mesenchymal cell is going to divide and produce an osteogenic cell. And that osteogenic cell is going to divide and produce an osteoblast. And that osteoblast is going to surround itself with matrix. And so is the next one, and so is the next one, and so is the next one. And notice what ends up happening here in this endosteum is we form a circular layer of matrix around the blood vessel. And what would we call a circular layer of matrix around the blood vessel?
Concentric.
There you go.
Concentric lamella.
You form a concentric lamella. And is there still periosteum in there? Yep. And does it still have mesenchymal cells? Yep. So it forms another concentric lamella and another concentric lamella until it is completely surrounded the blood vessel. With concentric lamella. And then what we have is we have formed an osteon with a central canal containing a blood vessel at the center. So notice, by incorporating the blood vessels, we form our osteons, we form our compact bone. So not only are we making the bone wider in width, but we are also forming our compact bone. We are also forming our osteons. Now, one last piece to the puzzle. As we can, yeah, I will save these pictures. But again, I think the pictures from your textbook do a nicer job of showing this, but I will save this. As we grow the bone in width, do the adult bones have a tiny medullary cavity and a massive amount of bone matrix in them? No. So as we expand the as we expand the bone in width, don't we also need to enlarge the medullary cavity? Yeah. And here in the endosteum, don't we have a special type of cell? And that special type of cell happens to have a big ruffled border on it and happen to have a bunch of nuclei that would allow it to be able to produce lots of digestive enzymes to break down the matrix and increase the size of the medullary cavity. What type of cell might that be? Osteoclast. Excellent. So while we are growing in width, our osteoclasts break down and enlarge the medullary cavity. And that is how we get our mature width of our bone. All right, let's look at the pretty pictures from the textbook and do it again. So notice, and again, here they do a nicer job of showing the different components to this. Here we have our periosteum with those immature cells, the mesenchymal cells, the osteogenic cells, making osteoblasts, making matrix, surrounding themselves with matrix and becoming osteocytes, and forming those circumferential lamellae. But we have to respect the boundaries of the blood vessels. So the matrix starts to expand around those, forming those periosteal ridges. As these ridges expand, the periosteal ridges are going to fuse together. And again, notice this does a much nicer job of showing how that periosteum is still contained within that space. So when it pinches it off, it forms a tunnel around the blood vessel, but that tunnel around the blood vessel still has a periosteum, although now it's surrounded by bone, so it's not a periosteum anymore. It is an endosteum, but it still contains those immature cells, so we can make new layers of matrix. And that's what happens. We start making layers of matrix that grow inward, those concentric lamellae, until we have the blood vessel completely surrounded inside of that central canal, surrounded by an osteon. And notice the same things happening right next to it, and so on and so forth. So in this fashion, we are both growing the bone in width, but also forming our osteons, making our compact bone. So really, growing in width is really two processes. It's how we get width to our bone and also how we form the compact bone. And notice also, we hinted at this before. If you think back to intramembranous and endochondrial ossification, In endochondrial ossification, that bone cuff and the compact bone occurred underneath the periosteum. That's where we form the compact bone. And in our intramembranous ossification, it wasn't until the periosteum formed on the outer surface that we then made that outer cortex of compact bone. We need that organized periosteum on the outer surface to be able to make compact bone. So not only can we grow it in width, but we need that periosteum to make our osteons. So we can incorporate those blood vessels, make them part of the bone, and make those osteons. All right. Questions on that? Excellent. Perfect. So we have now made bones and we have now grown bones. That allows us to go from balls of cells to little fetuses with bones. It allows us to go from baby-sized people to grown-up sized people. But I think pretty much everyone in this class is a grown-up sized people who is at their full grown-up sized people size. But that doesn't mean the bones get to take the rest of your life off. Bones are incredibly dynamic tissue. I can't emphasize this enough. Very dynamic tissue. It is constantly being broken down and remade. So there's a tremendous amount of remodeling that takes place on our bone. We're constantly breaking down old bone tissue and making new bone tissue. And of course, we could say making new bone tissue, breaking down old bone tissue, but what's the fun in that if we can give them fancy terms instead? And so that's, of course, what we do. The fancy term for breaking down old bone matrix is the process of bone resorption. And what cells would be involved in breaking down bone matrix?
Osteoclast.
Osteoclasts, absolutely. And remember also, we always have to think of the flip side of it. When we break down the matrix, releasing the calcium, where does that calcium go? When we break down matrix and we release that calcium, where does the calcium go? Yeah, blood, absolutely. Technically into the interstitial fluid, but once it's in the interstitial fluid, then it immediately goes into the blood. Because remember, what happens in the blood happens in our interstitial fluid as well. The other process, the process of taking calcium, taking it out of the interstitial fluid and therefore out of the blood, and depositing it into the bone on those collagen fibers, is the process called bone deposition. And of course, what cells are responsible for bone deposition? Osteoblasts. Excellent. We take calcium from the interstitial fluid. But again, if it's from the interstitial fluid, we're really taking it from the blood, because those two are pretty much interchangeable. All right, and we put it in the bone. So or into the matrix. Let's be consistent with our terms. So we break down the matrix, releasing the calcium into the interstitial fluid and blood, or we take calcium out of the interstitial fluid and blood and put it in the matrix. These are the two processes. So our osteoclasts are breaking down calcium, and our osteoblasts are building up calcium into their bone. And if your job is to maintain homeostasis, what should the relative rates of these two processes be? These are the easy questions. If our goal is to maintain balance and homeostasis, absolutely. They should be the same. The same rate that we are breaking down bone, we should be building bone. And as long as these are occurring at the same rate, the overall amount of bone density we have stays the same. So to maintain homeostasis, the rate of resorption and the rate of deposition should be equal. However, is it truly equal for your entire life? Well, I'm asking
No.
a question, so the obvious answer should be no. So what about as an infant, as an adolescent? What is the relative rates of resorption versus deposition?
Deposition is higher as an infant.
Yeah, as an infant, much more making of matrix than there is of breaking down. Remember, we talked about their bones aren't fully calcified yet. That's why they bounce like rubber balls, right? And if that kid rolls off the changing table, he's not likely to be hurt. On the off chance he does break a bone, it's pretty much healed by lunchtime, right? But what about grandma when she rolls off the changing table? Does she bounce? Does she heal by lunchtime? No, because as we become more elderly, right, what happens to the rates of resorption versus deposition? Yeah, deposition increases. Like we talked about, grandma can be drinking a whole cow's worth of milk, but if the rate at which her osteoblasts the osteoclasts are breaking down matrix is faster than the rate that she's making it, she's still going to be losing bone density. Now, it's still better to be drinking calcium than not drinking calcium, absolutely, because you want to have those resources, but it is still a concern that the rates of these do change. Our rate of making new bone changes as we get older, which is why it takes longer for us to heal from those types of injuries. Now, our goal is to maintain homeostasis, but there are three main factors that influence how successful we are at maintaining this homeostasis of our bone. What are they? What's one of the factors that can influence the rate at which you make big, strong bones?
Age.
I'm sorry, say again?
The age.
True. Okay, age definitely affects the rates of these, absolutely, but that's not something you can really control, right? What are some of the main factors that you can control that influence that?
Calcium.
All right, excellent. So yeah, nutrition, like getting enough calcium in those nutrients. I see exercise on there. And what's the third? So nutrition, exercise, and what else can influence these cells and what they do? We already talked about it. Hormones, there we go, hormones, absolutely. The three main factors that are going to influence our maintenance of homeostasis of our bones is going to be hormones, nutrition, and exercise. Let's talk about these three factors. Start with one of the easier ones first, right? Activity is vital for bone maintenance. All activity?
No.
What kind of activity in particular?
Weight bearing.
Yeah, exactly. We want to put stress on the bones. It is vital to put stress on the bones. Putting stress on the bones helps to maintain the strength and the density of our bone. If you are not doing weight-bearing activity, you break your leg or hurt your back or something like that, and you're laid up in bed for a period of time. Or the other place where it becomes a major, major issue is our astronauts in outer space. Without gravity, you can't do it's very difficult to do weight-bearing type of activities. And you can lose as much as 1/3 of your bone mass in just a few weeks of being in space. It's one of the major issues that that astronauts had when they first started coming back, and now they have a lot of elaborate specialized types of exercises that they try to do up there, which is what allows them to stay up there for months now instead of just days. The reason for this is that bone adapts to our stress. When you put a stress on the bone, it strengthens the bones, and it also aligns the osteons along that line of stress. So the bones become thicker and the bones become heavier. There are a couple really important implications to this. One of the places that you see this is in individuals who do asymmetrical type of activities. Probably the classic example of this are tennis players. Tennis players obviously do many two-handed strokes, but they do a lot more one-handed strokes. I'm not sure cashiering falls into the weight-bearing type of activity, not the same stress that you're putting on your arm with that strokes of the tennis racket. And they've actually done x-rays of both the dominant and non-dominant arms of tennis players. And the bones in the tennis players on their dominant arm are much, much thicker, much stronger as a result of that. That's definitely a good thing. It also, muscles, as they pull on it, enlarge the attachment points, right? If you happen to know a cyclist or know a soccer player, one of the things that I encourage you to do is to palpate their tibial tuberosity. What is the tibial tuberosity again?
That's the leg.
Yeah, it's on the leg, right below the kneecap, right below the kneecap. There is a bump right below the kneecap, and that bump right below the kneecap is your tibial tuberosity. One of the things that I encourage you to do is to find, if you're not if you don't have easy access to a soccer player or a bicyclist, the next time you're out on the street and you see a bicyclist going by, tackle them off their bike and palpate their tibial tuberosity. They won't mind at all. And what you'll see is those attachment points, especially in the runners, especially in the bicyclists, get quite pronounced as a result of that. Yeah, but in fact, it's the insertion point for all of your quadricep muscles. Yeah, tackle them on the grass. I mean, you don't want to be rude, but yeah, tackle them onto the grass at least. You don't want to hurt them. Excellent. But they have that strong attachment point, and these are definitely good things. However, one of the concerns that we have is that as you put stress on the bone, as we mentioned, it aligns the osteons towards them. So one of the things that happens is people will have a soft tissue injury. They will tear a muscle, or they'll pull a ligament, or something along those lines. And it changes their gait. It changes their stride. Right now they're walking with a cane or something like this. And as you're walking at a new angle, that is going to misalign the osteons. And you often see cases in athletes, as someone, like has a high ankle sprain, or they, strain their groin or something along those lines. And then they're dealing with that for a couple of weeks, and they come back the next week and they break a bone in their leg. And that happens because they've misaligned their osteons, and especially with chronic issues of people using canes or walking with a limp, it can become a serious issue where you can really make the bones more fragile by misaligning the osteons in them. Now, the question becomes, how does this occur? And the way it occurs is actually really, really cool. What happens is that we have that hydroxyapatite crystal in the bone. And when you have oh, wow, Kylie, you've had a rough week. 2 bones in your foot. That is not fun. Absolutely. Yeah. So again, one of those things you got to be really careful about how you're stressing your other bones as a result of that. Now, the reason this works is because of those hydroxyapatite crystals. When you compress the hydroxyapatite crystals, what happens is they produce a small electrical current, right? And that electrical current stimulates the osteoblasts, and the osteoblasts build more, right? Hopefully, most of you have experienced this. If not, it is a homework assignment that you have to do this weekend. One of the things, if you haven't done this before, you need to do, and now is a perfect time because everybody's got their Halloween candy out. So what you need to do is go to the store and buy wintergreen lifesavers. And then you're going to take one of those wintergreen lifesavers, and you're going to lock yourself in the bathroom with the light turned off, and then you're going to chew it. And why are you going to do that? Someone tell me why you do that. Come on, nobody's done that before? Nobody at Girl Scout at camp or something like that? Go ahead, Laura, what'd you say?
Makes sparks.
It creates friction.
Yeah, no, it makes sparks. Who said that?
Me, Allie.
There you go. Allie, excellent. Absolutely, right? It makes sparks. What happens with those wintergreen? So again, if you have not done this before, this is, call Grubhub or whatever it is, right? Instacart, have them deliver some wintergreen lifesavers to your house, right? That way it's stress-free and there's no worry that way. Take them and your whole family, go into the bathroom, look at yourself in the mirror, look at each other, and rapidly chew those wintergreen lifesavers. And when you do that, you will actually see sparks in your mouth, because what you're doing is you're compressing the crystals on those wintergreen lifesavers. And when you compress the lifesaver, it produces a spark. And that's exactly what happens with the bones. When you compress the cartilage, when you compress the matrix with that weight-bearing exercise, it generates an electrical signal, and that electrical signal stimulates the osteoblasts.
Could that also cause collateral ligament damage?
No, I mean, it's not that big of an electrical current. It's not like plugging yourself into a light socket. It's very local where that occurs. And again, you'll see that when you chew on it. It's not like sparks are flying out of your mouth like a dragon breathing fire or anything, but you do get tiny little sparks of light that will occur in your mouth. So it's not causing any issue that way. It's just compressing it. And again, as we learn how the body works, we use it to our advantage. While most of our bones are well vascularized, there are some bones that are not as well vascularized. The patella is a great example. Back in ancient times, there was a football player by the name of Jerry Rice, the greatest wide receiver to ever walk the surface of the planet, and he broke his patella. And so one of the things they ended up doing is they actually used electrical stimuli to stimulate the patella, right? They actually put an electrical current into his patella to stimulate it to try to heal it more rapidly. Sometimes in the wrist bones, they will put electrical stimulators on there because some of the wrist bones don't get as much blood to them, so they're slower to heal, and so we can put those electrical currents. And that's why I was curious, Kaylee, with your 2 broken bones in your foot, depending on which bones there are, they sometimes may put an electrical stimulation into those as well to try to facilitate and make the healing process occur more rapidly.
Can you use those, gosh, I cannot think of what they're called. Those two, you stick like a pad on one side and a pad on the other, and it goes through electric current for,
Yeah,
I think it's like blood stimulation. Tensing
right,
it.
or the things, they also have those things now that, you know, if you don't want to be bothered working out, you can eat chips and watch TV while you wear this thing around your waist, which stimulates your abdominal muscles to be able to do that. I don't think those things would produce a signal that would go deep enough to cause the bone to, I guess it would depend on where you put it. If it was a very superficial bone, like the carpal bones, you may be able to do something like that, but it typically is more a local thing. Remember, we don't want, we don't want to wrap something around and make the whole thing. We want to make it more focal. So typically they put electrodes directly into there. So again, I'm not encourage anybody who's broken their bones to go plug themselves into a light socket or anything like that. But it is something that can be done. Oh, wow. Tiny bones, the proximal phalanges. So yeah, those are small enough. Well, although I don't know if they would, because of their location being so distal, I don't know if they would do something like that. But again, they do it, like I said, for a lot of like the wrists, and you see it in the kneecaps and things along those lines.
What about like talking about exercise, like gymnasts, how they're generally shorter, like their growth is stunted. Can this be like too much?
So as we've talked about, putting stress on bones can stimulate the osteoblast. So if you think about it, if you're constantly doing those impacts where you're doing the running, you're doing the bouncing, you're doing all of the floor work, is it possible that that constant stress could cause their epiphyseal plates to close more rapidly? Yeah, something like that could occur. There's also a concern with that with weightlifting. If individuals start weightlifting at too early of an age, that also could cause a premature closing of the epiphyseal plate. So yes, that may be one of the reasons why.
I thought one of the reasons with gymnasts was they were exercising so much for puberty, so they weren't getting the estrogen and stuff to make their bones and muscles grow. That's why they were so short.
Well, and they generally don't go, like they have a later menstrual cycle.
True, and so what I will say to that is that one of the factors that influences when menstruation begins is the body fat. From a biological standpoint, what the female's body wants to say is, I am healthy enough to have children, to be able to grow and sustain a child within me. And one of the ways that the body shows that health is by having fat. Fat produces a hormone called leptin. And that leptin hormone tells the hypothalamus, all right, I'm ready to start menstruation. And it actually can increase, not increase, but it helps to stimulate menstruation process. So children that are very small in stature typically tend a longer delay in the onset of menstruation. And by the flip side of that, in some of these extreme athletes, some of these endurance runners that run Ironmans every other weekend, or they're doing one of those 48-hour races where they're just running 100 miles over a 48-hour period of time and all that, when they get down to 0.5% body fat, for some of them, they actually stop menstruating as a result of that. So it could also be related to that as as well, yes. All right. Excellent. Questions on this one? You guys are, got to be careful with all these broken bones that are going on, right? When you get to muscles, I don't want anybody going out and tearing muscles just to fit in with the organ system or the nervous system. No traumatic brain injuries when we get there either. All right. When we get to the digestive system in 431, if you want to go to the hot dog eating contest in New York, that you're welcome to do. But no more of these homeostatic imbalances. There's no extra credit for that, so be careful. All right, let's talk about another easy or straightforward one, and that is the nutritional requirements that are necessary for bone growth. Obviously, there is the need for calcium and the need for phosphorus. As we said, those hydroxyapatite crystals are primarily calcium phosphate. But there's also magnesium, there's also fluoride, iron, manganese. All of these are minerals that are directly related to the matrix. So obviously, we need the nutrients to make the matrix. Right, that is obvious, hopefully. So these are things that must be ingested, and if we're not ingesting them, we're not going to be able to make new bone. Calcium's the obvious one, but there's lots of macronutrients as well. But there are other nutrients that are necessary for bone homeostasis as well. One of the important ones is vitamin D, right? If I remember back to the integumentary system, where do I get my vitamin D again?
From sun.
Right, absolutely. From the skin, absolutely. Basically, you have a cholesterol in your skin that when exposed to sunlight for only about 10-15 minutes a day, they get converted into vitamin D. Vitamin D then, when activated, oops, there we go, becomes a hormone called calcitriol. And calcitriol's job is to tell the digestive system to absorb calcium. So again, you could drink a whole cow's worth of milk, but if you don't have that calcitriol in your body, most of that calcium will stay in your digestive system and will be released in your feces. To get your body to absorb that calcium, we need calcitriol. Now, right, the good news is it only takes about 15 minutes of sunlight. The bad news is between Corona and the smoke and TikTok, nobody's outside anymore. So luckily, they put vitamin D into our beverages now instead. So we get our vitamin D that way and can still get all the calcium we need from our drinks. But it isn't just vitamin D. Vitamin C is necessary as well, right? There was a time in world history where the Great Britain was the greatest country on the planet, right? The sun never set on the British Empire. They controlled a majority of the surface of the planet. And how were they able to do that? Because they had the greatest navy at that time, right? Yeah, turn the screen brightness, exactly. Greatest navy at the time. However, as great as their navy was, they were almost decimated by this horrible disease where the skin was peeling off their arms and their gums were bleeding and their teeth were falling out and their bones were breaking really easily, right? This horrible, horrible condition called scurvy. It almost decimated the entire British Navy. And how did they resolve it?
Taking what, vitamin C?
True, although, again, they didn't have pills they could take back then. So what did they do instead?
Maybe citrus, use lemon.
Yeah, they ate limes.
Oranges.
They ate limes, they ate lemons, exactly. They ate citrus fruits to get the vitamin C. Vitamin C, as it turns out, is vital for the making of collagen. That's why their skin was peeling off. That's why their gums were bleeding. That's why their teeth were falling out. Right? Yes, their teeth are still falling out. They have horrible dental care, exactly. But also, that's why their bones broke easily. Remember, to make bone matrix, we have to be able to deposit those hydroxyapatite crystals onto the collagen fibers. And if you can't make collagen fibers, you can't make bone matrix. So it is vital that we get our vitamin C to be able to make that collagen.
Question.
Yes.
Doesn't too much lemon break down the enamel on the teeth?
Well, so you are correct in that eating a lot of lemon or sucking on lemons, which is one of those things, you know, when you go to the restaurants and they have the little wedges of lemons in all the waters. My daughter used to love to suck on those all the time. Too much exposure to the citric acid of the lemon can affect the enamel of the teeth. So yes, in that case, it isn't the vitamin C that's causing the problem. It's actually the citric acid and that acidity. And the same thing with well, coffee for that matter. Other things have the ability to be able to break down the enamel that way. So yes, it's the acid of it that is the problem, not the vitamin C.
Oh, okay.
Notice so far, everything we've talked about is involving the matrix of our bone, but let's not also forget about our bone marrow as well. Things like vitamin A, vitamin B12, vitamin K are all vitally important for making bone marrow or making the cells associated with bone marrow. Vitamin K, for instance, vitamin B12 play a vital role in the making of red blood cells. Without B12, without K, we can't make red blood cells. And remember, our red blood cells only last for about 100 days. So we have to constantly be able to make that. And so having these other types of vitamins and other micronutrients are vital. So it's more than just having that glass of milk in the morning that is necessary for getting our bone density. All right, questions on that? All right, those are the straight.
I ask you one question regarding vitamin C.
Yes.
If a person does not want to eat like lemon, citruses like lemon, oranges, so especially the lemon, not the. So does it mean that the person doesn't need vitamin C and his body?
No, you get vitamin C from lots of other places. Broccoli actually has almost as much vitamin C as an orange does. So there's lots of places where we get these macronutrients from.
Tomato?
Sure. I actually don't know about tomato. That one I don't know. But I know there are lots of other things that have these. So it isn't just citrus fruit that you get it from. I'm sorry. Thank you for pointing out to me. JC said, so if you don't eat or drink dairy, then you need to get calcium from other things. Again, broccoli is one of these things that actually surprisingly has a lot of these things in them. There are other foods that have calcium in them. Some beans have calcium in them. So if you are vegan, there are other things that can be taken to make sure you get your calcium necessary. But you are correct. Those who go on a vegan diet do have a more challenging time getting all of the nutrients necessary to maintain the matrix. And so that can be a problem that way. So yeah, fruits, vegetables, beans, all of those things can have those as well. Cauliflowers, excellent, perfect. Yep, exactly. Eat a rainbow, right? Isn't that what you're supposed to do? All right, excellent. So these are the straightforward ones. Where things get a little bit trickier is when it talks about our hormones. Because again, as we talked about, we have this relationship between bone matrix and calcium levels in our blood. And as we talked about, these are not always in tune with each other. Sometimes they're antagonistic. So let's talk about some of the hormones that are involved in regulating our bone matrix. We already just talked about one of them, and I think I didn't put it on the list here, so let me cheat. And yep, perfect. Excellent. So since we just stopped it, there we go. Let's add it to the list here to make sure we have it. Calcitriol is again that active form of vitamin D. Right. We get it originally from the skin. The kidney then, skin is where we get the vitamin D, although it's actually activated in the kidney, the lungs, liver, stuff like that, but we won't worry about that process. So the skin for that. And as we talked about, its job is to increase absorption of calcium from the digestive system. All right, so let's throw that one on there because we don't want to forget that. And so that means I also need to erase this. So we talked about the sex hormones. The sex hormones, remember, as we talked about, the estrogens and the androgens, stimulate the osteoblasts. And remind me again, do they both stimulate them equally? No. Which one did it more?
Osteoblast.
No. Which sex hormone affects the osteoblast?
Androgens. Androgens.
Androgens do?
Estrogen.
Estrogens.
Estrogens, yep. Estrogens. Females are better. Estrogens have a bigger effect on the osteoblasts. All right. Now, if we think about this, then their job is to stimulate the osteoblast. So this would increase deposition. And if we're increasing deposition, there are two other impacts of that. We are increasing bone density. And we are obviously getting that calcium from the blood. So we are going to be lowering calcium levels in the blood. So those are basically the two main effects. So hold on, I need to move this out of my way here. Move that down there. Of our estrogens and androgens. They increase deposition, which increases bone density and lowers calcium levels in the blood. Excellent. So those are the hormones we've talked about already. Let us talk about a new hormone. That new hormone is called calcitonin. Calcitonin is produced by the thyroid gland. And its overall effect is to decrease calcium levels in the blood. And so it is going to encourage the removal of calcium from the blood and interstitial fluid. It's going to do this by basically having three targets. The first target is the bone itself, and more specifically, the osteoclast. For the osteoclast, what it is going to do is it is going to inhibit the osteoclasts. If we inhibit osteoclasts, then if you think about it, that decreases resorption. So we're breaking down less matrix. Right? Notice we're still making matrix at the same speed we were doing it before, but now we're breaking it down slower. The effect of this is we get an increase in bone density, because now we're putting more bone into the matrix, and it lowers calcium levels in the blood. All right, but that isn't the only target. A second target of calcitonin is the kidney. While you're sitting here in class, resting in your chair, 25% of your blood is being directed to your kidneys, because your kidneys' job is to filter your blood. In fact, you filter 200 liters of substances out of your blood during the course of a 24-hour period of time. Now, do you have 200 liters of extra fluid in your body? No. Do you produce 200 liters of urine during the course of a day? No, we'd all be in the bathroom doing this lecture if that was the case. Absolutely not. We reabsorb over 99% of that. But there's this massive filtering process that takes place. Well, what calcitonin does is it tells the kidney to stop reabsorbing calcium. It basically tells the kidney, hey, all that calcium you're normally bringing back into the blood, stop doing that. So what happens is there's more calcium in the urine, leaving the body, and it lowers calcium levels in the blood. Lastly, calcitonin inhibits the production of calcitriol. Remember, calcitriol is what absorbs calcium from our digestive system. If there's less calcitriol, then less calcium is absorbed from the digestive system. And if we're bringing less calcium in, then again, there's less calcium in the blood. So notice, if calcium levels get too high, calcitonin will help to bring these calcium levels back down. It tells the digestive system, stop bringing calcium into our body. We don't need any more. It tells the kidney, get rid of all that extra calcium we have. And it tells the osteoclast, stop breaking down bone matrix. We don't need any more calcium. So all of these effects help to bring blood calcium levels down. I've written that all out here, but we have it here as well. So let's cheat and erase that. So we can, oops, actually, let's not erase that. Let's cheat and move this. There we go. Over here. All righty. Here, I don't know if I can change the size of that anymore. Nope. Oh well. That'll work. Okay. So that is calcitonin. That's great if you have that nice big huge glass of milk for breakfast and you have lots of calcium in your blood and you need to get rid of it. But what happens if you have that Diet Coke and donut for breakfast? Are you getting all the calcium that you need? No. And so in those cases, we need to actually go to the bone, go to the kidneys, go to the digestive system, and get calcium that we need. To do that, we need another hormone. And that second hormone is parathyroid hormone. Conveniently enough, parathyroid hormone is produced by the parathyroid gland. Now it's small. There we go. And guess what? It also has 3 targets. One of those targets, assuming I can spell target right, is going to be the bone. And in this case, it is going to stimulate the osteoclasts. When we stimulate the osteoclast, that is going to increase resorption. That is going to lower bone density because now we're breaking down the bone matrix faster than we're making it. So it lowers the bone density, but it releases calcium into the blood. And that brings blood levels up of the calcium. It also targets the kidney. And it tells the kidney to reabsorb more calcium. Right? Tells the calcium, hey, all that calcium you were going to let go out in your urine, bring it back. We need it back. So obviously there's going to be less in the urine, which we don't really care about so much. The real key to this is that it brings back more calcium, so it increases. So let's say it this way. Yeah, we reabsorb more calcium, so that increases calcium levels in the blood. And not surprisingly, the third effect is that it stimulates production of calcitriol. So we bring in more calcium from our digestive system. Notice calcitonin and parathyroid hormones have the same 3 targets, and all 3 of them are opposite effects. One is going to bring calcium levels down, the other is going to bring calcium levels up. because as I've said many times and will continue to say through this and all of 431, calcium makes cells do wonky things. We need to keep very precise control of our calcium levels in the blood. So if it comes down to a choice between having the right levels of calcium in our blood and having big, dense bones, bones are going to lose every time.
Does long-term use of calcium channel blockers mess with the bones too, or is that just focused on the electrical impulses in the heart and stuff?
Great question. Yes, in this case, it isn't affecting the absorption of calcium. What it's affecting is the effect that that calcium can have on the heart. It limits how much can get into the heart and therefore affects the strength and of the contraction and the rate of the contraction. So it helps to moderate heart activity. Can help. It's an important. Helps to lower blood pressure, for instance, things along those lines. But it doesn't affect necessarily the bones. It just affects how it's able to get into the heart and affect the heart. All right, I have a pretty picture, two of them, that actually do a really great job of showing this. Let's go ahead and clear this and show this. And there's 2 points I want to make about this when we're looking at this. So again, here is what happens if our calcium levels are too low. If our calcium levels are too low, then our parathyroid hormone is released. It tells the kidneys to bring back more calcium. It stimulates the osteoclasts, so they break down more bone matrix, releasing the calcium into the blood, and it stimulates our calcitriol to tell the digestive system to absorb more calcium and bring calcium levels up. Conversely, if blood calcium levels are too high, we release calcitonin. Calcitonin tells the kidneys release the calcium, get rid of the calcium in the blood. Inhibits the osteoclast, so less calcium is released and more is deposited into the bone by the osteoblasts, and it stops calcitriol production, so less calcium is brought in from the digestive system. Now, there's one other point that I want to make for you. Notice as you look at this, look at what the low level is, 8.5 milligrams per deciliter. Look at what the high point is, 11 milligrams per deciliter. Now, you don't have to memorize these numbers, right? Are these numbers useful? Absolutely. These are the kind of things that you can use to woo women at a bar, right? Walk up to a woman at a bar, saddle up next to her and say, hey, did you know that the normal range of calcium in your blood is between 11 milligrams per deciliter and 8.5 milligrams per deciliter? And they just swoon when you do that, right? Now, are there numbers in this class that are going to be important? You are going to have to memorize? Yes, absolutely. These are not two of them. But what I want to show you is how incredibly narrow that range of normal calcium levels in the blood is. 11 milligrams to 8.5 milligrams is a tiny, tiny range. And so again, it should show you how vitally important calcium levels are in the blood. Maintaining that precise control of calcium, because calcium makes cells do wonky things. So we don't want to have too much of it. We don't want to have too low of it. We want to keep it in a very, very tight restrictive range. All right. Questions on that? Excellent. With that, we are done with everything we needed to do from a lecture standpoint. We are on to the bones and bone features now. So let's go ahead and take our next break. We will take a quick, what time is it now? 10:13. So we will come back at 10:28, and at 10:28, I will do a quick introduction to the rest of our axial skeleton, and then our groups will do their presentations. All right. So any questions on that?
Presentation on what, sir?
The group presentations. While some people did their the skulls, we have the rest of the axial skeleton that needs to do. So, so again, if you presented on Monday, you're off the hook. If you didn't present on Monday, today is your day to do that.
Yeah.
All right. So, yep. So we start at, like I said, 10:28, and at 10:28, we will pick up from there. All right. And since we'll be doing groups, I will not be recording the group presentations just like last time. So it is important to be here, be active, pay attention for these groups. All right. Questions on that? All right, and then after the group presentation, so even if you presented on Monday, obviously you need to be here to get the information from the rest of the groups that are presenting their material. But the other thing we want to make sure that we do is at the end of the presentations today, I will release the appendicular bone that you are going to be responsible for. We'll keep you guys in the same groups, and then I will release the handout that has the appendicular bones. I'm not going to do it before the groups because I want you focusing on what the groups today are presenting, but after that, I will give you that information. Sure, I'll tell you what. So what we'll do here, I'll tell you what. Well, let's go ahead and move it to 10:35. So we will restart at 10:35. And what I'll do is I'll break you guys into your groups. So anybody who needs to meet and talk will have the opportunity to do that during the break. And then we will restart at 10:35. All right. Questions on any of that? So that way we'll give a little bit more time because I get someone who asked that they want to meet in their group. So we will restart at 10:35 and I'll put you guys in your groups now. So give me a minute to get that done and then I'll put you in your breakout groups for those who need to prepare for today. All righty, excellent. So see you guys in, that's 20 minutes now.